Hello and welcome back to Science of Medicine. In this video, I want to talk about the electrical activity of the heart and how that impacts the beating and functioning of the heart. So it's convenient for us when we're learning the cardiac cycle or the sequence of events that happens from one heartbeat to another to break this into two pieces. This is a construct for our minds only. It doesn't actually occur. But the convenience of understanding the electrical activity and then understanding the pump activity before we integrate them makes it easier for us to learn. So I'm going to take it in those two steps. The electrical activity, which will be the focus of this video, and then the pumping activity, which will be the next. So if I look at the electrical activity of the heart, I find that I have several different pathways for conduction of electricity. You may remember that the myocytes themselves will have a potential difference across their membranes, but I also have an electrical activity centers within the heart that will allow for current to flow. And this is a small current. What I find that is in the right atrium, I have a group of cells that are intrinsic pacemaker cells. And these will fire at a certain rate, generally about 60 to 100 beats per minute. Now these can be modulated via the sympathetic nervous system to increase the rate, or they can be modulated via the parasympathetic nervous system to actually reduce the rate below 60 beats per minute. But this electrical activity will start with these SA node pacemaker cells and then propagate through the atria and come to a point at the atrial ventricular node, or the AV node. This is located between the atria and the ventricles, ergo the name. And there it's going to collect the signals and pause just a brief time to make sure that what is further propagated through the heart is a concerted signal. So once that pause has occurred, the signal will be conducted through the AV node down the bundle of His. That will then split into two portions. One that will go to the left side of the heart, that's called the left bundle branch, and the other that will go to the right side, that's called the right bundle branch. These bundle branches then go to the apex of each ventricle and fan out with the Purkinje fibers. And those Purkinje fibers then will also transmit that electrical signal. Now, along the way, these will cause depolarization of the myocytes and therefore contraction. Now, if I just had pacemaker cells in the SA node beating at 60 to 100 beats per minute, and I had a problem with those cells and they stopped working, or if I had blockage of the signal somewhere along the pathway, all of a sudden that's a fatal event. I die. I would have cardiac arrest. My heart would stop. So we have a set of backup pacemakers in a couple different areas. Right around the AV node, there is a set of backup pacemakers that if a signal is not received, they will actually beat around 40 to 60 times per minute, generally closer to 40, but we traditionally teach 40 to 60. And so if for some reason the signal from the SA node doesn't make it to them, then they will initiate contraction, albeit at a much smaller pace. And there is yet a backup to the backup within the Purkinje fibers. Now these are only there to try to keep you out of cardiac arrest, and they will beat at somewhere around 20 to 40 times per minute. And that is not going to keep you vertical. That's going to actually put you down on your back and be horizontal, where you're going to be doing nothing other than just simply living. But it's important to understand this process, and the only way that you can understand it is to actually understand the conduction system to begin with. Now it turns out that over 100 years ago, Getz had actually recorded this electrical activity of the heart by putting electrodes around the heart. And these can be done on the surface of skin or underneath the skin, in fact. And so I can think about this from an engineering standpoint as putting the heart, which is traditionally on the left side, placing that heart into the center of an equilateral triangle. And then I can look at different vectors or different views of the electrical activity of that heart. And that process of collecting that information will result in the construction of what's called an EKG or an ECG. Now, I need to have skin electrodes, surface electrodes, and I'll take the potential difference between differing two, and I'll get different signals from that. Now, it turns out that I can place these on. I have 10 different leads, that is 10 different electrodes that I'm going to place around the heart as well as on the limbs, both the arms and leg. And I'm going to take the potential difference between two of those, and I'll get different things. I'll have leads 2, 3, AVL, AVF. I'll have precordial leads, V1 through V6. These are important clinically because I can tell something about the anatomy of the heart and which blood vessels that supply the heart might have a problem in it. I'm not going to talk about that in this video, but there is much more information that is embedded within these. I'm going to talk about some very basic ideas as it relates to the conduction. So as I mentioned, I can take the difference in potential between any two electrodes, and I'll record the potential, which is shown here on the y-axis, versus the time, which is shown on the x-axis. And depending upon which two ones I actually take a difference between, it'll look a little bit different. And this will then lead to what I call a 12-lead EKG. What kind of information is embedded in this? So for a normal functioning myocardium, for a normal heart, I will have the following sequence, which is called sinus rhythm. If I'm plotting the potential, which is along the y-axis, versus the time, which is along the x-axis, I will have 60 to 100 beats per minute. I will have an initial upward deflection, which I will refer to as a P wave. And this represents the depolarization of the myocytes in the atria. Now, it turns out the right is before the left, but normally we don't resolve those unless perhaps the right atrium or the left atrium will be bigger, in which case I will see a little hump in terms of those. I have a segment between the P and the R wave, which is this big one going up, which is a PR interval. And that's just basically the time that it's actually taking to travel through the AV node. So I collect that bit of signal and it travels through the AV node and then goes down the rest of the conduction system. I have this fairly complicated looking series of peaks. I have a downward deflection after the P wave, an upward reflection, and then a downward again before I go back to the baseline. And this QRS complex represents the ventricular depolarization, as well as buried within it, the atrial repolarization. And I can tell a lot about what's going on from this QRS complex. I also have a QT interval, which is the time that it takes to actually depolarize and repolarize the ventricles. And I have another upward deflection called a T wave, and this T wave is the repolarization of that ventricle. So if I start depolarization of the ventricles here and go all the way out to the end of the T wave, that's my QT interval. That's important because there are many medications as well as poisons that will actually prolong this, that makes it more likely for a person to develop certain abnormal rhythms. You may have also heard about this ST segment, the little area right here between the S and the T waves, that it can be indicative, it can rise up or be depressed when there is blockage within blood vessels going to the heart and causes a type of myocardial infarction or heart attack called an ST elevation myocardial infarction. When looking at these PQRST waves, there are several ways of characterizing this. One of the ways is to look at the rate. The rate is simply the time that it takes to complete that cardiac cycle. And since you know P is when you're starting the depolarization of the atria, and the end of the T wave is when you're actually done repolarizing the ventricles, and then you go to the next P, you would have a way of doing this. For ease, we actually do, for regular rhythms, the rate as between the R and the R. And the reason for this is that back a long time ago when we used to record these potentials, the P waves were very, very small. We had difficulties distinguishing the signal from the noise, and so it was just more convenient to use the biggest signal, the R wave, to the next biggest signal. This only works if it's a regular rhythm, but the rate is how fast it's going, beating 100 times per minute, beating 180 times per minute, beating 20 times per minute. This is the rate. The rhythm comes in lots of different flavors, and I'm going to talk about several of them, and I'm going to relate these to the conduction system. So the first I've already alluded to and spoken about, called sinus rhythm. Sinus rhythm means that the signal is beginning in the SA node. It'll travel the expected way through the atria to the AV node, down the bundle of His, out the Purkinje fibers, and cause depolarization of the ventricles. So that means that if the rhythm is sinus, every P wave will have a QRST. So P, QRST, P, QRST, P, QRST. If the rate of this sinus rhythm is between 60 and 100 beats per minute, we call it normal sinus rhythm. If it's faster than 100 beats per minute, we call it sinus tachycardia for fast heart rate. And if it's less than 60, we call it sinus bradycardia for slower heart rate. There are other rhythms that can originate in the atria, but that do not originate in the SA node. These are atrial rhythms, and there are a lot of them. I'm going to talk about atrial fibrillation and flutter, but there are a number of other ones, and they are different from the sinus rhythms, as we'll see. I also have the possibility of having what are called junctional rhythms. The junctional rhythms come from the AV node, not from the sinus node. Come from the AV node, and they look different. And then there are ventricular rhythms where the signal originates from the ventricle, and these also look differently. In addition, I can have conduction abnormalities. I can have what are called heart blocks, which fortunately I call first, second, and third degree. I can have bundle branch blocks, both on the left and the right, and I'm going to just touch on the existence of accessory pathways, of which there are many that are important clinically. So as I mentioned, a long time ago, we used to record these EKGs on a strip chart recorder where the amplitude in the Y direction represented the voltage and the time was given on the x-axis. And so for one of these big boxes, it was 0.2 seconds to go there, and you could also get an amplitude peak here on this to say how many millivolts high it would be. Sometimes we count very quickly in terms of big boxes from an R wave to an R wave. This is the old-fashioned way that we would do it. We'd go to the first one, that would be a rate of 300, 150, 100, 75, 60, and so on. You can certainly do that, or you can simply say, oh, it's 5 boxes apart, therefore it took 5 times 0.2 seconds or 1 second to go for 1 beat. Let's talk about these various rhythms. So the first thing I want to talk about are sinus rhythms. Remember that the sinus rhythm originates in the SA node. If it is between 60 and 100 beats per minute, I call it normal sinus rhythm. So this first EKG, which shows potential versus time, is actually showing a sinus rhythm that is actually at a rate of 78 beats per minute. So it is a normal sinus rhythm. I look, I have P, Q, R, S, T. P, Q, R, S, T. So every P wave has a QRST associated with it. This is a sinus rhythm. And since the rate, this spacing, the time that it takes to get from one R wave to another is between 60 and 100, I call it normal sinus rhythm. This wave down here, this tracing down here, is also a sinus rhythm. I see a PQRST, PQRST. Every P wave has a QRST, so it's a sinus, but the rate is faster. It's actually 125 beats per minute. So I call this a sinus tachycardia. It's kind of like if you squeeze these together and made them faster. Similarly, if I use my same normal sinus rhythm strip up above, but I compare it with one where the rate is 50, now you can see these PQRST waves are spaced out, this is a sinus rhythm, but since it's less than 60, it's a sinus bradycardia. Now this is contrasted with other atrial rhythms. In this first set, the bottom tracing is sinus rhythm. So PQRST, PQRST, all these P waves look the same, but this top one is very different. Now I have this sort of squiggly, irregular baseline, and that squiggly original baseline is also causing for variations that I see between the spacing of the R interval. It's not a constant rate. This is called atrial fibrillation. There are no clear P waves. There are actually a series of these discharges, depolarizations. And so I'm getting these different foci in the atria that are causing depolarization. Another pattern is when I have atrial flutter. Atrial flutter shows this kind of sawtooth wave back and forth, and it's also irregular, just like atrial fibrillation. And this sawtooth pattern is very typical for atrial flutter. Now, one of the things that you get with both atrial fibrillation and atrial flutter is you don't conduct each and every one of these signals through the AV node, which is why you're getting conduction only at certain times. Now, in contrast to these, I have junctional rhythms. Junctional rhythms are when the signal originates in the AV node, or very close to the AV node. And this shows up as two different manifestations. The first is where I will have inverted P waves, but a normal kind of QRS width in terms of this. So you can kind of see this inversion here. Kind of see this inversion. I don't have a P wave out in front. Kind of this inversion here. And the second is where I have hidden P waves, because this P wave is actually sort of buried within this complex. But you can see here it looks beautiful, nice, but no P waves here that you can see. These are junctional rhythms. The width of the QRS is not widened. We'll talk about that in a second. Junctional rhythms arise in the AV node. Ventricular rhythms arise in the ventricles. So in this case, you can see several things that are very different. You still see no P waves, but now the QRS is very widened, and the T wave goes in the opposite direction as this QRS. And you also get this actually originated in the left side, and so you'll get these little bifurcation here, sometimes rabbit ear kind of looking. And so this is actually a type of ventricular rhythm, which is an escape rhythm, which is an accelerated rhythm. And sometimes you will see this transiently, hopefully transiently, after a person has had some kind of problem, major problem, like they've been in cardiac arrest. But this kind of widening of the QRS, this discordance, the direction between the QRS and the ST regions, and the absence of P waves are all clues that this is originating in the ventricles. Now, sometimes you'll get a rate that will be between the 20 and 40, where the backup Purkinje fiber pacemakers are actually the ones that are coming off on this. Let's talk about heart blocks. So they come in first degree, second degree, and third degree, and second degree comes in two types. So the first one is called first degree. And this is simply a prolongation of the PR interval to bigger than 1 box or 200 milliseconds. So you can see here, excuse me, is a P wave. And to go over here, this is more than one of these big boxes, 200 milliseconds. When this was originally investigated back more than 50 years ago on very healthy males, there was found to be no clinical significance of this. But it is a first degree heart block. Second degree comes in two types, and it's important to distinguish between these because they have very different clinical outcomes. The first type is type 1, or Mobitz type 1, shows the PR interval getting bigger. So here's PR, it's a little bit longer, and then I get a P wave, but I drop the QRST. This is a Mobitz type 1. Sometimes it's called Wenckebach. Mobitz type 2, or a type 2 heart block, I have a constant PR interval, but now I drop after a P the QRS-T. And this is something that needs to be dealt with very soon, because this generally will then degrade into third-degree heart block, which is requiring intervention. Third degree is where the P waves are doing their thing. They're polarizing, depolarizing, polarizing, and the QRS-T is also doing that. And there's no real relationship. So here's a P wave, and here's a P wave, and here's a P wave, and here's a P wave. P wave's buried here. And you have this, and then the QRS-T are just doing their own kind of thing. And that's because you're having two things. You're having the SA node doing what it's doing, but nothing's getting through, and then you're actually going on and doing these backup pacemakers that are doing their thing. And so third-degree heart block needs intervention. Typically, this is pacemaker dependent, where you will actually have to insert some sort of device that will allow you to beat and pace that rhythm. I mentioned also that you can have what are called bundle branch blocks. If you remember when we talked about the AV node and then going from the bundle of His, that it broke into a right side and a left side along the interventricular septum between the ventricles. And so if there's a block on the left side, it's called a left bundle branch block. If there's one on the right side, it's called a right bundle branch block. And what I want you to notice are basically for these blocks that there are patterns. So the left bundle branch block looks like this. In this case, I'm looking at the leads V1 and V6. And I see again the same kind of discordance, which I also had mentioned to you before, and a little bifurcation here. And the right bundle branch looks similar in terms of this. I get more of this up, down, up, what they call a RSR prime, or rabbit ears, again, V1 and V6. Now, I mentioned at the very beginning when I was talking about different rhythms that there exist accessory pathways. These are areas of the myocardium that conduct electricity, but they're not supposed to. And so one of the common ones, especially for younger people, is what's called AV nodal reentrant tachycardia, in which there's a little pathway between the atria and the ventricle that allows for a little loop to basically continue to stimulate the contraction. And so these are reentrant tachycardias. There are multiple different possible pathologic pathways that are there, Wolff-Parkinson-White being one of those that is well known and described. These are generally dealt with clinically by ablating them or basically killing the cells along these pathways. So I hope that I've given you an overview now of the electrical conduction pathways for the heart. In the next video, we'll talk about the pumping action. As always, if you enjoy these videos, please leave me a comment or a like, and you're welcome to subscribe so you know as I put up additional videos. And I'll see you soon.